Team Teleport, or the Targeted Extraction of Lithium with Electroactive Particles for Recovery Technology, is a collaboration between researchers and students at the University of Virginia and engineers at PowerTech Water focused on recovering lithium from geothermal brine at the Salton Sea. The teleport approach can be described in 4 steps using 3-unit operations, which can be integrated together to recover lithium from geothermal brine. The process begins with selective capture of lithium into an iron phosphate solid intercalation material, which is highly selective for lithium over other ions present in the geothermal brine. The intercalation material extracts lithium ions and decouples remaining purification processes, which include an electrolysis cell and final crystallization process to recover lithium hydroxide monohydrate product. The process starts with extraction of lithium from the brine using iron phosphate solid particles. Preparation of the intercalation particle packed bed begins by crushing iron phosphate particles. Less than half a gram of this highly selective material is needed per liter of geothermal brine. The particles are loaded into a column, which facilitates contact between the brine and the packed bed particles and affords control over pressure drop. The packed bed is prepared at a consistent bed height to promote reproducibility between experiments. The geothermal brine containing an iron-based chemical redox agent, which may already be present in the brine or included as a minor additive, is pumped through the packed bed. The redox agent drives reduction of the iron phosphate particles and the simultaneous intercalation of lithium ions into the crystal structure of the material. Following intercalation, the packed bed is rinsed with water to remove residual brine entrained in the packed bed. This water can be recovered elsewhere in the teleport process. For analysis and verification, the lithium iron phosphate particles can be removed from the column after intercalation, and X-ray diffraction, or XRD, can be used to verify the conversion of iron phosphate to lithium iron phosphate, as can be seen in these XRD data, where the 200 peak is a representative indicator of conversion between iron phosphate, or FP, and lithium iron phosphate, or LFP. Following the intercalation process, lithium can be released from the packed bed using an oxidizing agent. Let's take a look at the overall packed bed cycle. The packed bed starts out as iron phosphate particles, or FP particles. The geothermal brine is fed through the column, and this converts the FP particles to lithium iron phosphate, or LFP particles, leaving the brine depleted in lithium, and the brine at this point is re-injected into the well. The LFP particles are then washed using water. This water can be ultimately recovered, but is used to flush residual geothermal brine from the LFP particles. At this point, lithium can be removed from the LFP bed using an oxidizing agent, which converts the LFP particles back to iron phosphate, or FP particles, and this results in a lithium-rich solution which is passed to our electrolysis cell. The resulting FP particles are further washed with water to recover any residual lithium, and that solution is passed to the electrolysis cell as well. This leaves the packed bed in the iron phosphate form, ready for another cycle of lithium capture from geothermal brine. Ion chromatography is used to evaluate the selective capture and release of lithium from solutions containing high amounts of sodium. Next, we'll discuss the electrolysis cell. The lithium-rich solution released from the packed bed is passed to an electrolysis cell. At the anode, the spent oxidizing agent is regenerated and can be used for subsequent lithium release from a LFP packed bed. Lithium passes across a membrane in the electrolysis cell and combines with hydroxide generated via water splitting at the cathode, where hydrogen gas is also produced as a side product. The composition of lithium hydroxide solution leaving the electrolysis cell is determined using ion chromatography. Finally, the lithium hydroxide solution can be dewatered in a crystallization process where water is recovered elsewhere. The resulting lithium hydroxide crystals are recovered via filtration and imaged using SEM. The purity of the product crystals can be verified using XRD. In summary, Team Teleport uses a high-capacity solid particle packed bed to selectively extract lithium from brine and release it into an order of magnitude smaller solution with excellent selectivity against other ions as analyzed using ion chromatography. Extracted lithium is converted to lithium hydroxide via electrolysis as evidenced by pH and ion chromatography, while also regenerating the oxidizing agent used to release lithium from the packed bed. Finally, X-ray diffraction analysis of the crystallization product reveals a lithium hydroxide monohydrate product resulting from the ready-to-scale Team Teleport approach.